What are your DNA codes? Let's start
Um,
with it.
Thymine, adenine, guanine, and cytosine.
Excellent. So we're going to make them, we're just using the letters for today. T, oops, T, capital T, C, G, A. Good. Okay, now it's going to get harder. Gabe, what does T pair with in mRNA?
Doesn't it pair with A?
It pairs with A if it's going to DNA, but there are no. Yes,
Oh,
it
it's
does. Sorry, nope. You're right. No, you're right. You're right. Sorry, it's Monday morning. Um, my bad. Melina, what does C pair with?
G.
G. Good. Caroline, what does G pair with?
C.
Excellent. Jackson, what does A pair with?
T.
T if it was DNA, but there's no T in mRNA or any kind of RNA. So what would it be? T is replaced with
O U.
U. All right. Ava Daly, what does A pair for? So from mRNA to tRNA, A pairs with. Remember what Jackson just learned.
U.
Excellent. Ian, what does G pair with?
C.
Excellent. Adrian, C pairs with.
C pairs with G.
Excellent. And Lucy, U pairs with.
Would it be T?
It would be, except for there are no T's in RNA, and this is tRNA, so it's still RNA. So what would it pair with?
A.
A. Yep. And for some reason let's see if we can get it up there. Okay. So if I were you, I would highlight all of this because this is like the holy grail of this, because one of the things that we do continuously is is transcription like this, and translation back. So this will be super useful to you. This is like a great cheat sheet to keep around forever. So take a minute and see if you can do this transcription. Um, let's see, who do I want to? Abigail, when you use the DNA to mRNA, so we're going to go to mRNA because that's the only way it can go. What part of that key that you just made are you going to use?
The second part, like the DNA to the mRNA.
Exactly. So use the DNA to the mRNA, and I'm going to give you 3 minutes to do this. Go. All right, how you guys doing? You done? Can you put a green check mark in the responses if you are done? And great. All right, when you're done, make sure you put a green check mark in the responses. Okay, so even if you're not done, check the work that you did get done and fill in the rest. So T pairs with A pairs with U, A pairs with U. C pairs with G, G pairs with C, G pairs with C, A pairs with U. T pairs with A, C pairs with G, G pairs with C, G pairs with C, A pairs with U, T pairs with A pairs with U, A pairs with U, A pairs with U, C pairs with G, T pairs with A, and G pairs with C. If you are confused about how that happened, again, you're going to go back to your cheat sheet. T pairs with A, C on the DNA, a C pairs with a G on the mRNA, a G pairs with C on the mRNA, and an A pairs with U on the mRNA. Okay, cool. Let's keep chugging and talk about introns and exons. All right, go ahead and write those down and then we'll talk about them. Okay, so when we're talking about genes, the gene itself, as it is appearing in the DNA, is not actually the same sequence of amino acids that are going to go into the protein that you're going to build, which is kind of wild. Like, it's weird that this happens, and I'll tell you, like, scientific hypotheses for why that might be the case. But what it turns out is that there are parts of the code that are cut out. So there are parts that are going to turn into the protein, and turning into the protein is called being expressed. So the expressed part, that gives you your exons. So the DNA that is going to, and the mRNA that's going to be expressed are exons. And then the parts that are not going to be expressed, and sometimes scientists used to call this junk DNA, but it turns out that probably it does. There's very few things that don't do anything. So it might be a way that organisms can change their proteins in times of stress. It could be a number of things. It could be leftover from evolution, like now parts that you used to use but you don't use anymore. However, what happens is that those pieces are spliced out by enzymes and the exons are stuck together. And that's your mature mRNA, and that's what's going to leave the nucleus. So go ahead and write that part down. All right, can you give me a green check mark when you're done writing this down, please? Green check mark when you're done writing this down. Okay. So what happens next? The mRNA is going to leave the nucleus. All right, can you put a red X in the participants? Hold on, let me clear the participants. Can you put a red X if you in the participants section if you need more time? Now I'm gonna clear all feedback so you can. If you need more time, put a red X in the participants, and then take it away when you're done. Awesome. All right, I think this uses Flash, but we're gonna try it anyway. Hold on. Nope. Oh, nope, it doesn't. Okay. Oh, yes, it is. That's what I thought. Okay. Let's look at this. All right. I'm going to preview a bunch of these and find one that I like. I forgot that the one that I use and have used for years was Flash, and there's no Flash anymore. Sad. Okay, so let's go. So we're going to leave that there. We've done transcription. We made mRNA from DNA. And let's go back to home. And everybody, please open up your transcription POGL. And if you would unsubmit it so you can correct it, that would be ideal. All right, so I'll read to you while yours are loading. DNA is often referred to as a genetic blueprint. If just in case you missed it, everybody, we're on the front page. We're scrolling down to 325 transcription POGL, and we're opening it up. All right, again, story time. In the same way that blueprints contain instructions for conducting the construction of a building, the DNA found inside the nuclei of cells contains the instructions for assembling a living organism. The DNA blueprint carries its instructions in the form of genes. In most cases, the genes direct the production of a polypeptide, from which other more complex proteins, such as enzymes or hormones, may be constructed. These polypeptides and other molecules run the organism's metabolism and, in multicellular organisms, dictate what each cell's job is. So, what is the language of these instructions, and how do they how are they read and decoded by the cellular organelles? This activity will focus on the decoding of genes in eukaryotes. Okay, so you have a diagram as usual, and this is where the answers are, remembering that in a POGL. Okay, so where in the DNA in the cell is the DNA found? Let's see, where are my participants? Start at the bottom. All right, Wyatt, where in the cell is the DNA found? Wyatt. Okay, Wyatt, can you log out and log back in and see if you can get your audio to work? Val, where in the cell is the DNA found?
Has HS.
In
Her
the
symptoms were painful.
nucleus.
Yes. Excellent. Ooh, let's make. Okay. Where in the cell does transcription take place, Shauna? Hey, Shauna, where in the nucleus does transcription take place? Sorry, where in the cell does transcription take place? Nate, perhaps you can help Shauna out.
I have inside the nucleus.
Excellent. All right, what polymer is synthesized during transcription, Melina?
Pre-mRNA.
Good. Let's try again. Wyatt, let's see if we can hear you. What monomers are used to construct this polymer, and where are they found?
Is it nucleotides? Or
Yeah.
like the bases?
Nope, nucleotides is right.
Oh,
And where are they found?
I didn't get that half of the question.
Okay, well, see these nucleotides? Where are they?
The nucleus.
Yeah, remember that no answer is too simple. Just because you've already answered nucleus 2 times here, why wouldn't it work again? Shauna, according to Model 1, what enzyme is required for transcription? All right, we apparently are missing Shauna. All right, May, according to Model 1, what enzyme is required for transcription? May, can you hear me?
I can hear you. I just
Okay.
don't know the answer.
Okay, well let's scroll up and see. Does anyone know the ending for all enzymes? I think we've talked about it before. Okay, all enzymes end with ASE. So it's something that's doing something because that's what enzymes do, and it ends with ASE. So what is the enzyme? It was also in our notes today, May. And I'm pointing to it. Lucy, you want to give her a hand?
Was it, well, did she get the first half of the question? Like, I don't know where we are really. Like, I know what question we're on, but.
Yeah, what enzyme is required for, number 3, what enzyme is required for transcription?
RNA, I don't know how to pronounce the second word, but
Polymerase.
Yep.
Excellent. And the ASE ends all. So make sure that you're writing these things down if you didn't have the answer.
Walking club starts Thursday. Did you choose after school activities?
Okay.
What did you choose after school?
Refer to Model 1. What is the base pair rule for DNA strand matching an mRNA or an RNA strand? So let's see. Josh, what did you get for this? Josh, did you get this one?
Yeah, sorry.
It's okay.
Guanine pairs with cytosine, adenine pairs with uracil, and thymine pairs with adenine.
Cool. And let's do, just for the sake of continuity. Okay, so again, we are going to highlight this because this is like the thing that you need to be able to do. So highlight that. All right, good job, Josh. Jillian, compare this base pair rule with that of 2 DNA strands.
I didn't know exactly how to answer this, so I said that A pairs with the U.
Yes. So and that's different because in DNA, A doesn't pair with U, right? Right, Jillian?
Yeah.
What does A pair with?
T.
All right. Also, you could highlight that. That is important information to know. That's like a star circle thing. So let's highlight it. We'll pick a new color. Okay. Which strand of DNA contains the blueprint for the pre-mRNA? let's see. Where was I in my list? Julian has just gone. Ian, which strand of the DNA contains the blueprint for the pre-mRNA?
The template strand.
Yes, that works out fine. We'll call it that. Oops, that's not it. In what direction is the DNA molecule red? Griffin. Did you get this one?
I didn't really understand this one. I didn't know if it was talking about like. I don't really get it.
That's a good. I'm down with you not getting it. This is a complex concept that because the DNA is not the same on each side, they're like this side has a T and this side has a G and this side has an A and this side has a T and this one has a C and this one has a G. They are called anti-parallel and they have what they call a 5' and a 3'. So if you look at the template strand, the 3 is down here and the 5 is up here, and then this one the 3 is here and the 5 is here. I always say it's like your arms, so you have like a hand end and an elbow end, and instead of it being like hands to hands, it's like hands to elbows. So this is what anti-parallel means. They're parallel, but they have opposite. Your hands are on either side. And it reads from the hand end to the elbow end, if you will, or from the 5' end to the 3' end. And that's something that DNA is made that way. It's everything goes from 5' to 3'. And the prime is like a little apostrophe. And that has to do with where the carbons are. Don't worry about it. If you take biochem later, or if you take AP Bio, you'll learn about 5' and 3'. More than this. All right. In the DNA strand, the pre-mRNA strand, the DNA strand and the pre-mRNA strand are in anti-parallel. With this in mind, label the 3' and the 5' end of the pre-mRNA strand in model 1. So, based on that, here's the model. What side of this, is this the 5' end or the 3' end of the DNA molecule? This strand here. Are you on the 5' end of it or the 3' end of it? Jackson.
The 5'.
Yeah, it is. Let's see if I can get. So this is the 5' end. No, that's not a good color at all. All right, so that's the 5' end of the DNA. And so if this is the 5' end of the DNA, what does that make this end of the mRNA, Jackson?
The 3.
Yeah, good. I'm going to find a text color that works on that. Sorry about that. Nope. Oh, yeah, the white. Look at nope, not the white. Maybe the white if I red. Well, I don't know. 5 is the 3' end of the mRNA. Here, we'll put it down here so maybe there you can see it. Okay, label those on yours. That's the point of this. Cool. So the 5' end and the 3' end are always together. In which direction is the p-mRNA molecule constructed? So this is going to be backwards because you are reading the mRNA from the 5' to the 3'. The part that you're making, which part did you make first? You made the down here. This is the part that's already made. So if this is the 3', Jackson, what's this end? Since you're good at this.
Want to be the 5'.
Yep. So which side is made first? 5 prime. 5 prime. I give you the 5. I give you the 5. And guess what? This is too much detail. You don't need to know it. But it's good to like reach, get a little bit more concept. All right. Before the printing presses were available, books had to be transcribed in order to share the information on them. Consider the definition for transcription and explain why the process in Model 1 is described using that word. Do we have space for this? Oh, just okay. Let's see. Caroline, what does transcription mean?
I said you have to make a copy of the DNA.
Yeah, let's say write a good. Okay, we will leave it there for today. You do not need to, you don't really need to resubmit that. We'll actually probably resubmit it so your stuff gets saved. Save it or resubmit it, and we will keep working on this tomorrow. Cool? All right, have a great day. I will see you guys tomorrow.
Look at that.
Thank you, you
Thank
too.
you.
Bye.